Welcome to Layla's Science Hub. Today, we're simplifying graticules and staining. Everything you need to know for your exams and practicals made simple. First things first, how do we prepare slides for the light microscope? You can't just throw your sample on the stage and hope for the best. Here's the basic process. Place a thin slice of your sample onto a clean glass slide. Add a drop of stain if needed. We'll cover staining later on. Gently lower a cover slip using a mounted needle at an angle. This helps avoid trapping air bubbles. Finally, use blotting paper to soak up any extra stain or liquid. And just like that, your slide is ready to go. Now, cells are tiny, so how do we measure them? We use two things, the eyepiece graticule and the stage micrometer. The eyepiece graticule is like a tiny ruler inside your microscope, but its units don't mean anything until we calibrate it. This is where the stage micrometer comes in. It has a known scale, usually 1 millimeter with 100 divisions, equaling 10 micrometers per division. So how do we calibrate the eyepiece graticule? First, place the stage micrometer on the stage and line up its scale with the eyepiece graticule. Then, count how many graticule divisions fit into a known length on the micrometer. This gives you your calibration factor. Once you've got that, remove the micrometer. Place your specimen on the stage, count how many graticule divisions it covers, and multiply by your calibration factor to calculate its actual size. Let's look at an example. At times 100 magnification. 100 graticule divisions equals 1 millimeter, which is the same as 1,000 micrometers. So 1 division equals 1,000 divided by 100, which equals 10 micrometers. If your specimen is 15 divisions long, then the actual size is 15 times 10, which is 150 micrometers. Sometimes, microscope images give you a scale bar. This little line tells you the real-life length it represents. First, check what length the scale bar represents. Then use a ruler to measure the length of that scale bar on your image and convert units to the same as those on the scale bar, usually micrometers. Next, calculate the magnification using the IAM triangle, where magnification equals image size divided by actual size. Finally, measure the component in the image and rearrange the formula to calculate actual size. For example, you are given a cell that measures 8 centimeters and a scale bar that says 1 micrometer. You measure the scale bar as 4 centimeters, which is the same as 40,000 micrometers. Then calculate the magnification by doing 40,000 divided by 1 to get 40,000. So, to find the actual size, convert 8 centimeters to 80,000 micrometers and calculate the actual size by dividing 80,000 by 40,000 to get 2 micrometers. Next, let's talk about staining. Most cells are transparent, so stains are essential to add contrast. Different stains bind to different cell structures. This is called differential staining, and it allows us to distinguish between cell components. This makes it much easier to identify structures like nuclei, cell walls, or organelles under the light microscope. Here is a table of the stains you should know about. Exam tip: always check your units. Converting between units is very common in exam questions, so it's vital you learn these. It's an easy mark students lose. As well as this, make sure you are aware of the difference between magnification and resolution, where magnification is how many times larger an image is, whereas resolution is the ability to distinguish between two separate points.